Okay, the word glial cells comes from the idea that this basically means glue. So the full term is neuroglia, like you see up here at the top of the screen. Neuroglia. So it means literally nerve glue. These are the cells that bind all the neurons together. You have to imagine that the neurons are so specialized, they don't have time to do anything else except conduct nerve impulses. So all the daily housekeeping things that have to be carried out for a cell to live have to be carried out by helper cells, the neuroglia. Now these first several slides here are going to be dealing with neuroglia or glial cells found in the central nervous system. They are found only in the brain and spinal cord. The first of these are astrocytes. Astrocytes are the most abundant of all the glial cells. The word astrocyte means star cells, because again, if you take a look at their shape on my diagram over here, they do look like they have their stars, all the radiating branches coming out from them. Now, astrocytes, if you look at this one here, has some of its branches come off and connect to the neuron, here in yellow, and also to the capillary. So one of their functions is to brace the neurons. Give them a base of support so they aren't sliding all over the place. Now, they also will control the environment of the cell, of the neuron, because basically all that's happening here, nutrients that are coming through the capillary, through the blood, are going to be passing through the astrocyte and to the nerve cell. So it also forms kind of like a little filter. Anything bad that comes out of the capillaries can be absorbed here in the astrocyte and rendered harmless. This is what we call the blood-brain barrier, BBB. Blood-brain barrier. It's a way to keep the neurons from getting anything harmful. The blood-brain barrier, basically this astrocyte is part of it, is a way to keep toxins, in particular uric acid, which is produced from the breakdown of proteins, from entering the neurons and disrupting their function. Now remember, these cells, the astrocytes, are going to be found only in the central nervous system. Just like our next one, these are the microglia. The microglial cells are responsible for getting rid of any kind of junk, any kind of debris within the environment. So these are basically star-shaped phagocytes. Should bacteria get into the system there, part in the extracellular fluid, they're going to be responsible for digesting it. Remember, a phagocyte engulfs foreign particles and breaks them down. Also within the central nervous system, you have a special type of cell called the epididymal cells. Epididymal cells are very specialized. You find them only in the cavities of the brain. That means the ventricles. So you aren't going to find them just floating around every place. They're going to be in the ventricles of the brain, and they're going to be going up and down the central canal of the spinal cord. They look like this. This picture right down here is a beautiful example of how structure relates to function. Structurally, these epididymal cells have cilia on the surface. Now cilia, remember, are cell organelles designed for motion. And having cilia on their surface, they beat back and forth and move that cerebral spinal fluid throughout the central nervous system. They don't have any function to remove bacteria or any kind of other protection. They're just there to circulate the fluid. Now, the last of the cells, supporting cells of the central nervous system, are called oligodendrocytes. Now, oligodendrocytes have a close cousin that you're already familiar with. We talked about the nerve impulse conduction yesterday. We introduced you guys to Schwann cells, how they make a myelin sheath. Well, Schwann cells are found in the peripheral nervous system. Oligodendrocytes do the same thing within the central nervous system. Now, while like the Schwann cells, they're producing a myelin sheath for insulation and for a faster impulse conduction, there is a big difference between Schwann cells and oligodendrocytes. Besides their location, if you notice on here, one oligodendrocyte right here will spread out and cover several different axons. But a Schwann cell in the peripheral nervous system will only cover a small piece of one axon. That's a huge difference. Now, within the peripheral nervous system, there are two types of supporting cells that I'm going to ask you guys to know. We've already talked about Schwann cells on the previous slide and in our discussion yesterday with the action potential. Schwann cells will form each one of these little fatty sheaths here, these myelin sheaths, are composed of a Schwann cell wrapping itself around a small piece of the axon as it goes across. And remember, the purpose of the Schwann cells is to create the myelin sheath that speeds up nerve impulses. Now, the other cells in the peripheral nervous system are called satellite cells, because as they cover the cell body like this. When you look at them in an actual slide like this, notice the little black arrow there. They look like little tiny moons. Well, that's why they're called satellite cells. Now, the job of the satellite cell is to insulate and protect the cell body. So I've always told my classes, think about the satellite cell as like an overcoat. Let's protect the cell body from rapid changes in the environment. Because let's remember, the peripheral nervous system is outside of the brain and spinal cord. And brain and spinal cord are very well protected and very well maintained. If we're talking about going outside the central nervous system, like into our fingers and toes and other parts of our body, it can be more subject to kind of rapid changes. So I think the satellite cell, a good analogy, just call it like an overcoat, insulating the regular cells. So this